Hello friends, in this video I'll be talking about the regulation of glycolysis. Okay, so by definition, the glycolysis is defined as the conversion of this 6 carbon containing glucose into 2 molecules of 3 carbon containing pyruvate. Okay, and the process of glycolysis is independent of oxygen. That means there is no absolute requirement of oxygen for glycolysis to happen. Only after the pyruvate is formed, okay, and the fate of pyruvate is decided by the availability of oxygen or inavailability of oxygen. Okay, so there are two different respirations which we know, that is aerobic respiration, aerobic and anaerobic. Aerobic respiration and anaerobic respiration, anaerobic respiration. Okay, and after the pyruvate is formed from the glycolytic pathway, okay, if the pyruvate undergoes during aerobic respirations, the fate of pyruvate is TCA cycle. It's TCA cycle. Okay, the process of glycolysis, it happens in cytoplasm, and in presence of oxygen, the fate of pyruvate is it enters into the mitochondria and undergoes TCA cycle, which is amphibolic pathway, or citric acid cycle, which is a amphibolic pathway. And in case of anaerobic respiration, the fate of pyruvate is this pyruvate is converted into lactate. The pyruvate is converted into lactate. Okay, so generally the meaning of anaerobic respiration is during the process or during this anaerobic respirations, and this anaerobic respiration, it happens during vigorous exercise. Okay, one pathway for the anaerobic respiration is it happens during the vigorous exercise. And another thing, okay, there is cell that solely depends upon this anaerobic respiration, and that cell is RBC, because this RBC, it lacks mitochondria. Okay, so there is no aerobic respiration that can happen during the process. Okay, so the fate of pyruvate is decided by availability of oxygen or inavailability of oxygen. Okay, now coming to the regulation of glycolysis. So there are 3 steps in glycolysis that are regulated. So the first step that is regulated is conversion of glucose into glucose 6-phosphate. Okay, and the enzyme catalyzing this reaction is hexokinase, which uses ATP and also the magnesium ions for their catalytic activity. Okay, and this enzyme, it is regulated by the concentration of glucose 6-phosphate. Okay, the increasing concentration of glucose 6-phosphate exert a negative feedback on the hexokinase enzyme. Okay, so first regulatory step of the glycolysis is conversion of glucose into glucose 6-phosphate. Okay, next is the pyruvate kinase enzyme. This pyruvate kinase enzyme is the last enzyme of the glycolytic pathway, okay, that converts phosphoenol pyruvate into pyruvate. Okay, right? And this enzyme is regulated by the concentration of ATP inside the cell. That means the ATP, it inhibits the pyruvate kinase allosterically. It inhibits the enzyme pyruvate kinase allosterically. Okay, now one of the most important enzyme of the glycolytic pathway is the phosphofructokinase enzyme. Okay, so this phosphofructokinase enzyme converts fructose 6-phosphate into fructose 1,6-bisphosphate during the glycolysis. Okay? And this PFK enzyme, it is regulated by the concentration of fructose 2,6-phosphate. It is allosterically activated by this product, which is fructose 2,6-bisphosphate, okay, and it is inhibited by ATP. It is inhibited by the ATP. Okay. Now I'll be talking about from where this compound it is formed. Okay. So this PFK enzyme, okay, has another isoform which is PFK2, which is PFK2. This PFK2 enzyme has a dual activity. It has a kinase activity as well, and it has a phosphatase activity as well. It has a phosphatase activity as well. Okay. So during the glycolysis, when there is abundant supply of fructose 6-phosphate, when there is abundant supply of fructose 6-phosphate during the glycolytic pathway, or when there is abundant glucose present inside the cell, the kinase activity of this PFK2 converts this fructose 6-phosphate into fructose 2,6-bisphosphate. And the increasing concentration of this fructose 2,6-bisphosphate is an allosteric activator of the enzyme PFK1 that is involved in the glycolytic pathway.